Hello everyone, today we gonna study about roots, about dicotyledonous and monocotyledonous roots. So let's start from dicot. Well, we all know that dicot basically a group of angiosperm that produces an embryo with two cotyledon. And here is another group that produces an embryo with a single cotyledon. So there is basically two groups, one is dicot and another one is monocot. Now let's talk about anatomy. In dicotyledonous root, we can see the epidermis in the outermost layer, and this is a specialized parenchymatous layer with the multiple unicellular root hairs, and this layer is completely devoid of cuticle. Okay, now inside the epidermis we can see cortex, and that have multiple layers of parenchyma cells. Also, you can see some intercellular spaces between the cells. Now the next to the cortex there is a single layered area called endodermis, and in this area cells are barrel shaped and there is no intercellular spaces between the cells. And the most important thing about the endodermis, the tangential as well as the radial walls of the endodermal cells have a deposition of water impermeable waxy material called suberin in the form of a stripe known as Casparian stripe, and it produces root pressure. Okay, then the inside of endodermis we can see another single layered area called pericycle, and from this area initiation of lateral roots and secondary meristem take place. Finally, the centralmost part or area is known as stele, and it can be divided into two parts. Number one, vascular bundle, and there are usually 2 to 4 xylem and phloem patches. These are radially oriented, and the xylem is exarch in type according to the position of protoxylem and metaxylem. And in between the xylem and phloem there is parenchymatous layer called conjoint parenchyma tissue, and the central part of the stele is known as pith. It is generally reduced in dicot. So it is generally reduced in dicot. So this is all about dicot root. Now monocot root is approximately similar with the dicot root except in the area of pericycle and stele. Here the pericycle is parenchymatous just like the dicot, but it can only initiate lateral roots. They cannot form any part of secondary meristem. So they cannot form any part of secondary meristem. This is very important. And within the stele, there are usually more than 6 bundles of xylem and phloem. So here we can see more number of vascular bundles than the dicotyledonous root. So you should remember there was 4 and here is 6 number of vascular bundle present in the stele. Okay, you should remember this, there was 4 and here is 6. Now also the xylem is exarch in type and the orientation is radial, just like the dicotyledonous root. Now the most important thing about the monocot root that they lack secondary meristem, so that is why they do not undergo any secondary growth. This is very important and asked frequently in the exams. Monocot root, they lack secondary meristem, so that is why they cannot undergo any secondary growth. And the last thing is about pith, which is large in size and formed by the parenchyma cells.